You know that feeling, right? You wake up, maybe there's a little tickle in your throat, slight headache. You're not properly sick, but you can tell your body's, well, it's fighting something off.
Yeah, there's definitely an internal skirmish going on.
But have you ever really stopped to think, how does it do that? What incredible systems are working away behind the scenes, often without us even noticing?
It's fascinating, isn't it? A silent, constant battle. And today, we're going to pull back the curtain on that world.
Right.
Especially for you as a first-year medical student, we're doing a deep dive into the immune response, your body's amazing defense system.
So the goal here is to give you a clear roadmap.
Exactly. Complete with memorable anchors, metaphors, things to help you not just understand these vital concepts, but really retain them, recall them easily.
Okay, great. We'll explore how innate and adaptive immunity work together, the key cells, the whole coordination that keeps you healthy.
Sounds good.
So, to kick us off, here's our first anchor question, something to think about as we start. What's the very first line of defense your body uses against invaders? Before any, specialized cells jump in.
Good question.
Hold on to that thought. We'll definitely circle back to it.
Yeah, it's foundational, really. Puts everything else into context. Shows how much happens even before the alarm bells ring, so to speak.
Okay, so let's start right at the border.
Yeah.
The absolute frontier. Your natural barriers. Your body isn't just sitting there waiting for things to get in, is it?
Not at all. It's got these formidable defenses already set up, always there.
Like castle walls.
Precisely. Think of them as the castle walls and moats. Immediate things, physical barriers, chemical barriers designed simply to block entry. Your first major hurdle for any pathogen.
And these aren't just passive walls, right? You've got the obvious physical ones like skin, it's tough, selectively permeable. And then the cilia in your respiratory tract, like tiny brooms sweeping things out. The gut lining, too.
Right, the mucosal linings are huge. But here's a key thing. The cells making up these barriers, the epithelial cells, they're not just passive bricks in the wall.
Oh, what else do they do?
They actively produce these powerful antimicrobial peptides. They're called defensins, little chemical weapons, essentially.
Wow, okay, so physical and active chemical defense right there.
And speaking of chemical barriers, you've got things like lysozymes. You find those in saliva and tears.
What do they do?
They're enzymes that specifically break down bacterial cell walls. Pretty neat.
Yeah.
And then there's pH. Think about the stomach, super acidic, the urinary tract, the vagina, also low pH environments.
Which makes it hard for many microbes to survive.
Exactly. It's just inhospitable, an environmental deterrent.
It's amazing how many layers there are right at the start. And you mentioned if a breach does happen, those epithelial cells.
Yeah, they don't just give up. If something gets through, they start producing signaling proteins, things called cytokines and chemokines.
Like sounding the alarm.
That's a perfect way to put it. They're the castle's alarm bells, attracting other immune cells, activating them, getting ready for the next phase.
Which brings us right back to that first question we asked.
Ah, yes.
The very first line of defense.
It's these natural barriers, the skin, the mucus, the chemicals like lysozyme, the pH. They stop the vast majority of things before they even get a chance. That's your immediate frontline defense.
Incredible. So much protection just built in. But, okay, let's say something sneaky does get past the walls and moats. What happens then?
Right. That's when your innate immunity kicks in. This is the body's always-on rapid response team, ready to go instantly.
And the key thing about innate immunity is?
It's nonspecific. That's crucial. It doesn't care which specific bug it is, just that something foreign and potentially dangerous is there.
So it reacts the same way regardless of the invader.
Pretty much. And it's incredibly fast. We're talking minutes. But, and this is vital for you to remember, it has no memory. It treats every encounter like the first time. Starts from scratch.
Okay, fast, nonspecific, no memory. But how does it even know something's wrong if it's nonspecific?
That's through inflammatory signaling. It's the body shouting HELP. Basically, the innate cells have these scanners, these receptors called Pattern Recognition Receptors, or PRRs.
PRRs. Got it. What do they recognize?
They're looking for specific molecular signatures. Two main types you need to know. First, PAMPs.
PAMPs.
Pathogen-Associated Molecular Patterns. Think of them as signatures of invaders.
Yeah.
Things found on microbes, but not our own cells. Bits of bacterial walls, flagella proteins, viral DNA or RNA, stuff that screams foreign.
Okay, so PAMs are on the bad guys. What's the other type?
DAMPs, damage-associated molecular patterns. These are slightly different. They're distress signals released from our own cells when they get damaged or die unnaturally. maybe due to trauma or infection.
Ah, so the body can tell if it's an external threat or internal damage.
Exactly, and recognizing these PAMPs or DAMPs is key. When a PR, like the important Toll-Like Receptors, TLRs, binds to one, activation. The cell switches on and releases a flood of inflammatory mediators, things like histamine, prostaglandins.
And that's what causes the visible signs of inflammation we see.
Precisely. This leads directly to the 5 cardinal signs of inflammation. You'll see these in patients constantly.
Okay, let's list them. Number 1.
Heat and redness. That comes from increased blood flow. The blood vessels widen, that's vasodilation, more blood rushing to the area.
Makes sense. Flush skin around a cut. Number 2.
Swelling. The blood vessels also become leaky, increased vascular permeability. Fluid and cells leak out into the tissue. That process is called extravasation or sometimes diapetesis.
Okay, swelling. The number 3 must be
Pain. Partly from the pressure of the swelling on nerves, but also some mediators directly stimulate pain receptors.
ouch. And number 4.
Sometimes tied to pain and swelling, you get loss of function. Hard to use a joint that's swollen and painful, right?
Absolutely. Heat, redness, swelling, pain, loss of function. The 5 signs.
And while it feels bad, this whole inflammatory response is crucial. It's not a mistake.
It's the defense mechanism in action.
Yes. It brings more blood, which means more immune cells, to the site. And it allows those cells to get out of the blood vessels and into the tissue where the problem is. It's a coordinated call to arms.
Right. So who are these frontline soldiers of the innate system that rush in?
Good question. First up, the phagocytes. Cells that literally eat things.
Like Pac-Man.
Kind of. Top of the list are neutrophils. They're often called the first responders. Made in the bone marrow, rush to the site of inflammation really fast.
And they eat invaders.
They do phagocytosis, yes. They're also granulocytes, meaning they have granules filled with potent chemicals they can release. Very effective.
Okay. Neutrophils. Who else?
Then you have macrophages. They start life in the blood as monocytes. You can spot them by their kidney-shaped nucleus.
Monocytes in blood.
Then they leave the circulation, remember diapetesis, and mature into macrophages once they're in the tissues. They become specialized for that location, like Kupffer cells in the liver or microgliae in the brain.
And they eat things too.
Oh yeah, big eaters. Phagocytosis, cleaning up debris, killing pathogens. But crucially, they also act as antigen-presenting cells, ATCs. They show bits of what they've eaten to other immune cells.
Connecting innate to the next phase.
Exactly. And speaking of APCs, we have the dendritic cells, also from myeloid stem cells, also great phagocytes. But their main job, their superpower, is being really, really good APCs.
So they're like intelligence officers.
That's a great analogy. They capture the intel. The antigen then travel to lymph nodes to present it, briefing the adaptive immune system. Super important link.
Okay, neutrophils, macrophages, dendritic cells, any others in the innate team?
We should mention the other granulocytes briefly. Mast cells involved in allergies release histamine. Eosinophils and basophils roles in fighting parasites and also in allergic reactions. They all carry chemical weapons in their granules.
Got it. And one more important one.
Ah, yes. The natural killer cells, or NK cells. These are lymphocytes, but they're part of the innate system.
What makes them special?
They can recognize and kill virus-infected cells and tumor cells directly without needing prior activation or antigen presentation like the adaptive cells do. They're like innate assassins doing surveillance.
Wow. Okay, so the innate system is fast, uses inflammation, has these phagocytes, APCs, NK cells. It seems pretty powerful.
It absolutely is, which leads us nicely to our second anchor question.
Right. If innate immunity is so fast and effective, why do we even need this other system, the adaptive immunity, which you mentioned is slower? What's missing from the innate response?
Think about that for a moment. What crucial element isn't there?
Okay, let it simmer. Because the innate system is remarkable, but as you say, it has limits. And that's where adaptive immunity comes in, the specialized SWAT team, you called it.
Exactly. And its key features are almost the opposite of innate. It's very specific. It targets one particular pathogen or even one specific part of a pathogen with incredible precision.
But it's slower.
Yes. Much slower initially. It takes days to really ramp up the first time it sees something new. But, and this is the game changer, its superpower is memory.
Ah, immunological memory. Yeah. That's why you don't usually get chickenpox twice.
Precisely. The adaptive system remembers invaders it's encountered before, and if it sees the same one again, the response is much faster, much stronger, much more effective. That's the foundation of long-term immunity and vaccines.
Okay, specific, slow initially, but with memory. Who are the elite forces here?
These are primarily the lymphocytes, two main classes. T lymphocytes, or T cells. They start in the bone marrow, like most blood cells, but they mature and get educated in the thymus. That's the T in T cell.
Educated. What do they learn?
They learn to distinguish self from non-self, essentially. Very important training. Then they circulate, patrolling the secondary lymphoid organs like lymph nodes and the spleen, looking for their specific antigen.
And there are different types of T cells.
Yes, two main functional types you absolutely need to know. First, the CD4 plus T helper cells. Think of them as the coordinators.
Why coordinators?
Because they're central to orchestrating the whole adaptive response and even boosting the innate response. They secrete chemical messengers, cytokines, that help activate and direct other cells like macrophages and B cells.
Okay, crucial helpers.
But here's a key detail. They can only see or recognize antigens when they're presented on a specific type of display molecule called MHC class II, or MHCII.
MHCII for CD4 plus helpers.
Got it. And the other type.
The CD8 plus T cytotoxic cells. These are the precision killers or the assassins.
Okay, what do they kill?
They directly kill our own body cells that have become dangerous, mainly cells infected with viruses or cancer cells. They recognize specific foreign antigens displayed on the surface of these troubled cells.
How do they kill them?
They induce apoptosis, which is programmed cell death, a clean, controlled kill. They do this using weapons like perforin, which punches holes in the target cell membrane, and granzymes, enzymes they inject to trigger the self-destruct sequence.
Wow, precise and deadly. And what do they recognize? Not MHCII. No, CD8 plus cells recognize antigens presented on MHC class I, or MHCI. That's the other main display molecule.
Okay, CD4 plus sees MHCII, CD8 plus sees MHCI. That seems important.
It's fundamental to how the whole system targets the right threats.
Right. So those are the T cells. What about the other main lymphocyte?
The B lymphocytes, or B cells, they mature in the bone marrow. That's the B.
Makes sense.
B cells have receptors on their surface, a bit like antibodies, that can often bind directly to a foreign antigen. They don't always need an APC to show it to them first, although APCs and T helpers are usually involved in activating them fully.
And when a B cell gets activated, what does it become?
It differentiates into two main types of cells. First, plasma cells. These are incredible little factories. Their sole purpose is to churn out huge amounts of antibodies.
The antibody factories.
Okay.
And the second type?
Memory cells. Just like T cells can form memory cells, B cells do too. These are long-lived historians that stick around, ready to mount a rapid response if the same antigen ever shows up again.
Memory again. Super important. So how does this activation actually happen? You mentioned antigen-presenting cells, APCs.
Yes, antigen presentation is critical. This is where innate cells like macrophages, and especially those dendritic cells, bridge the gap to adaptive immunity.
They capture the pathogen.
Capture it, break it down inside, and then present little pieces of it, the antigens on their surface. And they use those MHC molecules we mentioned as the display case.
Right, the MHC display cases. Remind me which is which again.
Okay, MHCI is found on almost all nucleated cells in your body. It mainly presents endogenous antigens, bits from inside the cell, like viral proteins if the cell is infected, or abnormal proteins in a cancer cell. It signals, kill me, to CD8 plus T cells.
Okay, MHCI is for internal problems. Signals to CD8 killers.
Generally, yes. And MHCII is found mainly on those professional APCs, macrophages, dendritic cells, and B cells. It presents exogenous antigens bits from things the cell has eaten, like bacteria that were outside. It signals help coordinate a response to CD4 plus T helper cells.
MHCII is for external threats. Signals to CD4 helpers. Got it. That distinction is key.
Absolutely. And there's one more layer of safety. Co-stimulation.
Co-stimulation.
Yeah. For a T cell to become fully activated, it usually needs two signals from the APC. Signal 1 is the specific antigen binding to its T cell receptor via the MHC. Signal 2 is a separate co-stimulatory molecule on the APC binding to a receptor on the T cell. Think of it as a double handshake.
Why the double check?
It's a safety mechanism. It ensures T cells only get fully activated when there's a genuine danger signal, indicated by the APC being activated enough to express co-stimulatory molecules, preventing them from accidentally attacking healthy tissues.
Makes sense, a fail-safe. So this activation leads to the actual responses.
Exactly. Let's look at the antibody response, also called the humoral response. This is mainly driven by B cells.
Okay.
So B cell binds its specific antigen, it internalizes it, processes it, and presents it on its own MHTII.
Right. B cells can be APCs too.
Then, a CD4 plus T helper cell that recognizes that same antigen presented on the B cell's MHCII comes along and provides the crucial help co-stimulation and cytokines.
The helper cell helps the B cell.
Precisely. That fully activates the B cell. It starts dividing, rapidly proliferating, and differentiating into those plasma cells.
The antibody factories.
Yep. And they start pumping out millions and millions of antibodies specific for that original antigen.
And antibodies. They don't kill directly, right? You said they were like flags.
Exactly. Antibodies are Y-shaped proteins. They circulate and bind to the pathogen or toxin they're specific for. This can neutralize toxins, block viruses from entering cells, or a really important function, opsonize the pathogen.
Opsonize. What's that?
It means coating the pathogen with antibodies. This makes it much, much easier for phagocytes like macrophages and neutrophils to grab onto and eat the invader. It's like putting handles on a slippery ball.
Ah, okay. Antibodies tag for destruction. That's the humoral side. What about the T cells doing the direct killing?
That's the cell-mediated response, primarily driven by those CD8 plus T cytotoxic cells.
The assassins.
Right. They patrol, looking for any body cell that's displaying a specific foreign antigen, like a viral peptide, on its MHCI. Remember, MHCI is on almost all cells.
So if a cell is infected.
If it's infected and showing the wrong flag on its MHCI, the CD8 plus cell recognizes it, binds, and delivers the kill signal inducing apoptosis using perforin and granzymes. It's a direct cell-to-cell takedown. Very precise.
Okay, wow. Humoral for extracellular threats via antibodies. Cell-mediated for intracellular threats via cytotoxic T cells.
That's a good summary, yes. Though there's overlap and cooperation, that's the main division of labor.
Which brings us back finally to that second anchor question we asked. Why do we need the adaptive system if innate is so fast?
And now the answer should be clear.
It's the specificity in the memory, right?
Exactly. Innate immunity is fast but blunt. Adaptive immunity is slower initially, but incredibly precise, targeting only the specific threat. And critically, it remembers. Without that memory, every cold would be like the first time you ever got it. No long-term protection, no vaccines would work.
Can be constantly vulnerable.
Constantly. So they aren't separate systems, really. They work together in this beautiful, coordinated symphony.
How does that coordination work?
Well, the innate system initiates and informs the adaptive. Those dendritic cells especially are the crucial link. They capture the antigen at the site of infection, process it, travel to the lymph nodes, and present it to the naive T and B cells, essentially saying, here's what we're dealing with.
They're the messengers.
The intelligence couriers, yes. And then the adaptive system directs and amplifies. Once those T helper cells are activated, the cytokines they release don't just help B cells make antibodies, they also massively boost the killing power of innate cells like macrophages, make them angrier, more effective.
And the antibodies help the innate cells too.
Definitely. By opsonizing pathogens, antibodies make the phagocytes' job much easier. It's a powerful feedback loop.
So that inflammation we talked about earlier, the innate response.
It acts as a bridge. It draws all the relevant immune cells, both innate and adaptive, to the site of infection. It creates the battleground where everyone can work together effectively.
You can almost visualize it on an infection curve, can't you?
You really can. That initial phase, maybe feeling a bit off, the incubation,
Yeah.
that's often the innate system trying to contain things.
But then when you get properly sick.
That's often when the adaptive response is kicking in, clearing the pathogen. And then the memory ensures you recover fully and are protected long-term during convalescence. This partnership also helps resolve the inflammation properly, preventing chronic issues.
It's just an incredibly efficient and balanced system.
Yeah.
Speed, precision, memory, cooperation.
Truly remarkable when you break it down.
Well, this has been an incredible deep dive through the body's internal security force. We've gone from the castle walls, the natural barriers, to the rapid response team, the innate system with its inflammation and phagocytes.
Right.
All the way to the specialized SWAT team, the adaptive system with its T cells, B cells, antibodies, and that crucial memory.
Hopefully you now have a really solid conceptual map, understanding how these two arms work together to protect you. We aim to give you those memorable anchors, the castle, the teams, to help you retain this complex information.
You've charted this amazing defense system, figured out the key players, the signals, the critical interplay, and just to leave you with something to mull over.
The final thought.
We've seen how carefully the immune system identifies and eliminates threats. But what happens when it gets it wrong? When this highly tuned system mistakenly identifies something harmless, maybe pollen or peanuts, or even your own body's cells as a dangerous invader.
Yes, the consequences of friendly fire.
Exactly. What could be the consequences of that kind of misidentification? How might understanding that shape how you think about disease and treatment? Keep exploring, because the immune system, well, it holds many more fascinating secrets.